Yo, welcome to my lab. I got about 3 minutes to tell you about my research, how I'm way up in that. I work in Wollongong now from Sydney in Australia, investigating cancer of all types of paraphernalia. You see what makes a cancer deadly is how it can spread. Might start in your lung, then it's a tumour in your head. This is called metastasis, it's quite deadly. 90% of cancer patients die this way readily. We're trying to understand how tumours do this to stop metastasis and get a cancer to dismiss. One pathway is leading your body astray, helping a tumour grow and spread to leave you in dismay. It's quite a mouthful to say, man, but what the hey, it's known as the plasminogen activation pathway in a normal body. Helps a fetus grow, shape in the womb, build an artery, stops blood clots, repairs your wound. In the tumorigenic system, not the same benevolence gets hijacked by a tumour, creates unpleasantness. Cancer is intelligent, it needs replenishment. It has to get a blood supply to increase its eminence. Conscript paths, triggers, angiogenesis gets a tap feed on your blood. You might not see the end of this. Now the tumour cells escape in the leaky vasculature, making satellite tumours that are hard to endure. It's a tricky puzzle, prob sinking. How can we stop it? Well, PAS has this inhibitor. Part 2 could unlock it. Part 2 stops the breakdown of extracellular matrix, binding to its target protease, UPA. Yeah, it's irreversible. Gets taken up inside the cell, which means the cancer cells can be targeted quite well, cause the UPA receptor that helps tumour spread is overexpressed in tumour cells. And now you see where I'm headed. We're making a pro-drug, kinda like chemotherapy, and it's actually a natural protein in your own body. So we'll grow heaps of bacteria culture to express it. Leaders and leaders sweating just to get it. Then I got it, there we go, conjugated to some peg, purify it up, characterized by mass spec. Then get me that toxin from this periwinkle lad. Do a bit of chemistry, a few moles on the pad, then characterize again. We hope that drug's clean, so we can target specific tumors and know what I mean. We got that peg, cause it makes drugs a little bigger. Doesn't get filtered by the kidneys or the liver. Spends the time in circulation, patiently waiting to bind to UPA and start interrelating. Inside it's taken, the endosome starts breaking up the linkages. Now the drugs be making its way to the nucleus to start devastating. Turbulence like scaffold for new cells emanating. So we're all geneticists and cell biologists, doctors and chemists. Picture lots of sense, and it's evident that we're gonna bring some difference to cancer patients upon this residence. That's pretty much it. Gotta get back to my Ph.D. and just figured I'd tell ya how do we be doing it up in the lab, in the scenes behind us. Catch you later. Thanks for watching Fresh Science.
Peace.